Hello everyone and welcome to my channel. Here we are going to discuss about the general characteristics of the actinomycetes. Classification of actinomycetes. Actinomycetes are classified under a domain of bacteria. They are classified under a phylum Actinobacteria. It has a order Actinomycetes, family Actinomycetidae. Introduction. The actinophages, these are nothing but the viruses which can cause an infection to the actinomycetes. The actinomycetes, the member of family of actinomycetes are range from harmless bacteria to the pathogenic one, and these pathogenic bacteria or pathogenic actinomycetes are resistant to the number of antibiotics. Some important genera are listed here. These are Actinomycetes, Nocardia, Actinomyceta, Septomyces and thermophilic Actinomycetes. Now about the habitat where they can be found or where they live. Actinomycetes are a diverse group of bacteria. They are present everywhere in earth. Predominantly, they are found in a soil where they are acting as a degrading organisms. They are able to degrade some recalcitrant compounds like chitin and a cellulose. Recalcitrant compound means the compound which does not get decomposed easily. For example, the hydrocarbons, plastic, these are the recalcitrant compounds. They are able to degrade the such recalcitrant compound like chitin and a cellulose. For this activity, they require a higher pH. That means they are active at a higher pH. We know that the fungi are also ability to decompose the recalcitrant compounds, but they are active at a low pH. After a first rain, soil got a earthy or a musty odor. This earthy or a musty odor or a smell is because of the production of a geosmin. The geosmin is nothing but a carbohydrate like compound which is produced by a soil actinomycetes. Almost all the species are a common cells of a oral cavity and tonsils of the animal. Some of the actinomycetes are a normal flora of mouth and female genital tract. Morphology. The actinomycetes are a diverse group of organism or diverse group of bacteria. They can be found everywhere in a nature. Gram negative in nature, non-motile, non-sporing, non-capsulated. The shape of actinomycetes is straight, curved or pleomorphic. Pleomorphic means the organism which shows a phenomenon of a pleomorphism. Pleomorphism is a characteristic when the organism are growing, it changes its shape during the growth. The cells of actinomycetes are arranged in a chains or sometimes arranged in a branches, forming a branching filaments. The cell morphology of the actinomycetes is resembling toward that of the mycobacteria and a corynebacteria. We know that the mycobacteria, these are the bacteria which are causative agent of the tuberculosis and leprosy. The corynebacteria are the bacteria which are causative agent of diphtheria. The cell structure of actinomycetes is related to the mycobacteria and corynebacteria. The actinomycetes produces a mycelium The mycelium is nothing but it is a network or it is a tangled mass of hyphae. The morphology of Actinomycetes shows these are unicellular like a bacteria. As well, they produce a mycelium, and this mycelium is nonseptate and having a slender in nature. Nonseptate means number of cells are arranged in a one row and there is no any formation of septate. One cell shows a presence of one nucleus. The cell wall of Actinomycetes does not contain chitin and cellulose. We know that the cell wall of fungi contains chitin and cellulose. Actinomycetes produces a hyphae and a conidia. At the tip of hyphae there is the presence of conidia and a sporangia, and this characteristics is similar toward the fungi. Certain Actinomycetes whose hyphae undergo a segmentation and resembling to a bacteria, both morphologically and physiologically. Here means we can say that the Actinomycetes are morphologically resembling to the fungi, and in a cellular organization they are resembling toward the typical bacteria. In this image, there is a stained cells of Actinomycetes. How the mycelium get formed? The spore after germination forms a germ tube. This germ tube get divided and redivided into a hyphae. Further, the multiplication of this hyphae forms a such tangled mass or a network or a hyphal network called as a mycelium. Why the Actinomycetes are not considered as a fungi? Here are a differences in between fungi and the Actinomycetes. The Actinomycetes are non-motile filamentous gram-positive bacteria which are grouped into a class Actinobacteria. Whereas the fungi are a group of microorganisms which includes a single cell and complex multicellular organisms such as yeast, mushroom and molds. Actinomycetes are prokaryotic in nature, fungi are eukaryotic in nature. The cell wall of Actinomycetes shows a presence of petrolatum, whereas the cell wall of fungi contain chitin. The filaments of Actinomycetes are smaller, filaments of the fungi are bigger. Guanine plus cytosine contained in a DNA of Actinomycetes is less than fungi. The guanine plus cytosine bases in a DNA of fungi is more. Although the Actinomycetes are not considered as fungi and grouped into a bacteria, the Actinomycetes shows us some similar characteristics toward the fungi. These are Actinomycetes and fungi are filamentous in nature. They produces a mycelium. Both of them produce spores. Both of them are a good decomposers of recalcitrant products, and both of them produces antibiotics. Some characteristics are similar to the bacteria. The Actinomycetes and bacteria are prokaryotic in nature. They don't have any membrane-bound organelles like endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplasts. Such kind of membrane-bound organelles are absent towards both Actinomycetes and bacteria. Both have a murin or a petrolatum in their cell wall. On a solid media, both the organism produce a colony. Both bacteria and Actinomycetes produces a endospores. They occurs in an environment as a part of normal microbiota. Both of them can act as a pathogen. The next cultural characteristics or a growth characteristics of the Actinomycetes. The Actinomycetes are anaerobic in nature or they are microaerophilic. Microaerophilic means the organism which requires a low concentration of oxygen for respiration. The Actinomycetes are anaerobic or microaerophilic bacteria and grows well in the presence of 5 to 10% of CO2. The optimum temperature is 35 to 37 degrees Celsius. The selective media for the growth of Actinomycetes is brain heart infusion agar or brain heart infusion broth. We can make a use of thioglycolate agar also, and this thioglycolate agar contains rabbit blood. The incubation time for the growth of Actinomycetes is 3 to 4 days or it gets extended to 1 to 2 weeks. The colony on an agar media of Actinomycetes may be rough, pigmented with a chalky appearance. In this image, there are some colonies of Actinomycetes on an agar media. Clinical significance, as these are a normal flora of a human being or animals, they can be found in cavities of humans and other animals. The Actinomycetes, first time shown by a scientist Hertz, a qualitative agent for actinomycosis or a lumpy jaw of an animal or Actinomycetes. It was given or invented by a scientist Hertz. The Actinomycetes causes infection to a thoracic or abdominal cavities. Actinomycetes are qualitative agent for meningitis, endocarditis or genital infections. For a culture of Actinomycetes, we can make a use of selective media like blood agar, brain heart infusion agar, glucose broth or a thioglycolate broth. Incubation time, they can be incubated aerobically or anaerobically for 14 days. One species from Actinomycetes, that is Actinomyces israeli. After growth of this Actinomyces israeli on a thioglycolate medium, they forms a spider type of colony. The spider type of colony are resembling to a molar teeth. It is about the growth of Actinomyces in soil and in a nature or in an environment. Below this, here are some rich of nutrients by Actinomyces. The PCD after a programmed cell death, such kind of programmed cell death of cells of Actinomyces releases a nutrient in a soil environment. Also, during a growth, the basal cells which are present in a soil produce antibiotics. The aerial hyphae having a number of cells after growth, and after chromosome segregation and a separation, there is a maturation of individual spores. The spores get dispersed into the environment, and after germination of a spore, the spore leads to formation of a vegetative cell, and this vegetative cell into a soil produces a mycelium. This is a reproductive cycle of the actinomycetes. These are the colonies of the actinomycetes on a SCA medium, that is a starch casein agar. And one most important thing about the actinomycetes is that the number of antibiotics are produced by actinomycetes. The antibiotic like kanamycin, rapamycin, ascomycin, amphotericin B or rifamycin, these are produced by the genera of Streptomyces, and they are active against a fungi, bacteria. Out of all the antibiotics, 99% of antibiotics are from Actinomyces origin. Thank you. If you like the video, subscribe my channel and share it.